important area of immunology for many reasons, for historical reasons and for practical reasons. So what is the historical aspect? Well, what we call infectious disease or infectious diseases have been with us throughout history. If you look at history in different countries, there are periods that have been marked by epidemics or pandemics of infectious diseases. Sometimes they've been so vast that they have influenced the life of people in a grand scale. Here I have a painting that refers to basically what is called the Black Death epidemic, which happened in Europe in 14th century, and it was caused by an important disease. Sure. Oh, sure. And it was caused by a disease which was named plague. More specifically, it was named bubonic plague. Plague is a bacterial disease which happens in people after they get infected with a bacterium named Yersinia. And it's named bubonic. The word bobo in Latin, it means cucumber. It means cucumber, cucumber. Why cucumber? Well, this is a disease which is caused by fleas, F-L-E-A. What is a flea? Arabic word for flea? F-L-E-A. What is F-L-E-A? Flea in Arabic. F-L-E-A. Barghouth. It's a tiny insect that bites people, and it can infect us by biting tiny things, and it transmits Yersinia to us, usually from animal reservoirs like mice or rats. They have the Yersinia, the flea bites them, and it then bites us. And then if it bites on the feet, the Yersinia is transmitted to the inguinal region, lymph nodes. If it bites on the hands, it will be transmitted to the axillary region, and then lymph nodes will become big, so big that you can see them from outside. This is an unusual case of lymphadenopathy, and for that reason they've been calling it bubonic plague, referring to cucumber, tiny cucumbers. This is the Wikipedia page for that, and actually the picture is in here too.
Victor,
So, yes.
Do you mean the flea bites the human and make this disease?
Yeah. So fleas are the vectors. They transmit the disease from mice and rats, from rodents to human beings. Yes, yes. So that is bubonic plague. And for that reason, it's been called, the reason it's been called bubonic plague is because of these big lymphadenopathies. It's a deadly disease. And it has a story behind it. It's a sad or interesting story depending on your perspective. You know, Mongols, when they attacked eastern countries, western countries, this was Mongolia. So Genghis Khan was the first Mongol warrior or commander who led Mongol army towards the Iranian Empire at the time, and then he took Bukhara and then Merv and Nishabur and some other cities. Later generations of Mongols came all the way to Baghdad, and that's when they destroyed Beit al-Hikmah, or House of Wisdom. So they came all the way here. So that was the first group of Mongols, or one group of Mongols. The other group of Mongols went and captured the land north of the Caspian Sea, like today's Kyrgyzstan and Kazakhstan and those countries. They went all the way to this location. This is the story behind it, this location. This is a peninsula that you hear it these days. It's named Crimea or Crimea. Like Ukraine, Russia, they have dispute over it. There was a city in Crimea that the Mongol army led siege to. The name of the city was Kaffa. They circumscribed the city, but they couldn't enter because it had huge walls and there were people inside who were resisting. And a lot of those people were Europeans, Christian merchants from Italy and other countries. So they continued the siege for 1 year until something bad happened for the Mongol army. What was the bad news? for the Mongol army, plague appeared there. Mongols knew about plague very well. Everybody knew about plague very well. Like us, we know about coronavirus very well because we've experienced the pandemic. They knew about plague very well. And they knew that it's a contagious disease. It spreads. So when they told the commander that we have some plague patients, he was very scared. He said that we need to go back. We need to retreat. Because if we remain here, the entire army will be decimated. But before we go back, we do something to the people inside the city. They've been infected by plague. We're going to make them be infected with plague. How can we do that? Well, they had these catapults using to throw stones and stuff inside the city. They took the body of the dead people, put them in the catapults and threw them inside the city. So bodies were probably they had fleas, and the fleas were contaminated. So we don't know how many bodies they threw, maybe tens, maybe hundreds. And people inside the city were infected. This is the first documented example of a biological warfare. You use microbes to defeat your enemy. They didn't know about microbes, but they knew that plague is contagious. What happened later? Well, people from the city, they were Europeans. They went all over the Europe and contaminated the entire continent. And that was the beginning of Black Death epidemic. Killed almost 40% of European population at the time. So this is an important example of an infectious disease. But it's not the only important example. The guy here in the middle is Napoleon. And as you're seeing, his army are around him. But the army is in disarray. This is after Napoleon attacked Russia. They went and captured Moscow. But then they had problem providing food there for their army, so they had to retreat. The army was 500,000 people, 500,000 soldiers. It was named Grand Army. It's said that when Grand Army came back to France, from 500,000 people, only 45,000 were still alive. Some of them had died from lack of food. The rest of them had died from an infectious disease that was a nightmare for armies. It was named typhus. Typhus is transmitted by lice. Lice, L-I-C-E, is like mice, mouse, mice, louse, lice. What is the Arabic word for louse? Louse. L-O-U-S-E. L-O-U-S-E. It's another insect. Small, tiny, but bigger than fleas. L-O-U-S-E.
Hamlet.
Amla?
Hamlet.
Camel, it is a plural. Camel.
Camel. Okay, camel. Lice. Lice. The agent is a rickettsia. It's a sort of bacterium that is transmitted by lice. Typhus. This is another example of an infectious disease that is transmitted through water. No insects here. Through water. And it's the back of a journal in Paris at the turn of 20th century. It's named cholera or vaba, cholera. It enters the body by contaminated water, and it leads to dehydration. You lose water through severe diarrhea. It's another example of a deadly infectious disease. But the disease which has its name most commonly associated with immunology and vaccination is the smallpox. So the previous diseases, they were bacterial diseases. Typhus is bacterial. Cholera is bacterial. Plague is bacterial. This is a viral disease by viruses. And it leads to big blisters in the skin. This is another deadly disease that have happened many times. Unfortunately, we don't have it anymore because of vaccination. So these are historical aspects or historical examples of infectious disease. But we have infectious diseases today. The coronavirus epidemic wasn't one example of them. So we need to know about immune response against infectious agents. Why? Because if we know about immune response, we can protect ourselves. In addition to that, the way that immune response happens against an infectious agent determines the clinical manifestations as well. So it's important for prevention. It's also important for diagnosis of the disease. So there are three factors which influence the immune response. The first is the type of the pathogen. Is the virus? Is the bacterium? What kind of bacterium? What kind of virus? Is it the fungus? Is the worm? So that's important. The other part is whether it's a systemic infection. Systemic means it's all over the body. Or it targets specific organs like lung, or the brain, or joints, or gastrointestinal system. But the third factor is even more important. Where is the pathogen living? Does it live outside cells or does it live inside cells? Is it extracellular outside cells or intracellular inside cells? So what I'm going to do is to bring the type of microbe here. We have four major types of, instead of microbe, I better say infectious agent, because some infectious agents like worms, they're big. They cause infections, worm infections, but they're big. So we have bacteria, viruses, fungi, and parasites. By parasite, I mean things like worms or amoeba, for example. I bring them to the left side of the page, and then I put extracellular and intracellular there to have some examples in here. See what we have in here. Bacteria, most of the time they live outside cells, like E. coli that causes diarrhea, or pneumococci which cause pneumonia. But we have some bacteria like mycobacteria which live inside cells, like Mycobacterium tuberculosis, for example. It causes tuberculosis. How about viruses? Well, viruses have to live inside the cells. They cannot live outside cells. Fungi, they usually live outside cells, but we have some examples like histoplasma. It's a fungus that lives inside cells. And for parasites, worms, these are worms. Well, they are too big to live inside cells. But we have this, which causes malaria, why mosquito bite, malaria, which lives inside cells. So I'm going to use this table to categorize and explain immune response. But before doing that, we need to further separate intracellular. There are two types of intracellular, and there are two types of extracellular as well. Intracellular, I have one example in here. This is a mycobacterium. This is a virus. Mycobacteria, like Mycobacterium tuberculosis, enter our body through air by inhaling. They go to our lungs and they get phagocytosed by macrophages. Imagine that this is a macrophage. They phagocytose and put it inside something which is called the phagosome. Most bacteria die inside phagosomes, but mycobacteria don't. They live there. They like it there. They proliferate there. They kill the macrophage. They come out. They get phagocytosed by another macrophage. They live there. They kill it, and they keep going on and on until they lead to the disease named tuberculosis. This is an example of an intracellular pathogen. On the right side, I have some viruses. Imagine that these are hepatitis viruses, and this is a hepatocyte, liver cell. They go inside hepatocyte, they proliferate there, they kill the cell, they come out, they infect the next one, they kill it, they infect the next one, they kill it, until the liver damage happens. This is also an intracellular pathogen. But the one on the left side is different from the one on the right side. The one on the left side, it's inside the vesicle, it's inside the phagosome. The one on the right side is floating in the cytoplasm. And it's important for immune system whether you're inside the phagosome or whether you're floating in the cytoplasm. So I need to put two areas in here, intravesicular and intracytozoic. Both of them are intracellular. How about extracellular? We have two areas in here too. Sometimes extracellular means at the surface of the body, the surface of the skin, or the surface of mucosa. This is extracellular. Sometimes skin is penetrated or mucosa is penetrated and they go inside blood. or interstitial space. This is extracellular, this is extracellular, but two different types of extracellular. So now I can put some stuff in here. Mycobacteria live in here, inside vesicles. But rickettsia lives inside cytoplasm. Rickettsia, one of the diseases is typhus that killed Napoleon's army. Viruses are almost always inside cytoplasm. Fungi, if they are inside cells, usually they are inside vesicles. How about parasites? Well, Plasmodium is inside cytoplasm, but we have other parasites like Leishmania which are inside vesicles. Yes.
Which one is more dangerous? The cytoplasm or the...
Oh, all of them can be dangerous. All of them can be dangerous. So we can't say, we will see that. All of them can be dangerous. Yeah, yeah. Depending on the pathogen, they can be different. How about extracellular space? Well, sometimes I have an E. coli that lives at the surface of the intestinal epithelium and causes diarrhea. Sometimes it can penetrate and go deep. A streptococcus can cause pharyngitis by living at the surface of epithelium, but it can also go deep. Viruses are here. For fungi, usually they are at the surface of epithelium, like Candida, which lives at the surface of epithelium, causing candidiasis or thrush. Dermatophytes that infect the skin. They can be inside blood, but it's not very common. And so is the case for worms. A lot of worms live at the surface of epithelium in the gastrointestinal tract, inside gastrointestinal tract, at the surface of epithelium. But sometimes some of them might go deep. So this table is going to help us categorize immune response. How are we going to use this table? We have innate response and we have adaptive response. Innate response is mostly phagocytes. So for phagocytes, it doesn't matter. If it's at the surface of epithelium, they can have access. But if they come inside, they will phagocytose it. If they are inside cells, they cannot perform phagocytosis. But if the cell dies and the stuff come out, they can perform phagocytosis. But for B lymphocytes and T lymphocytes, the story is different. B lymphocytes can protect extracellular space, but they cannot protect intracellular space. Why? Because B cells produce antibodies, and antibodies don't have access to this region. Antibodies do not go inside the cells, they are outside the cells. So if there is something outside the cell, antibody can bind to it. But if it's inside the cell, they can't beat it. So this is the story that we have for T cells and B cells. One thing to keep in mind is that even though we have intracellular pathogens, these intracellular pathogens, in order to get to their destination, they need to go through the extracellular space first. Like if it's a virus that wants to live inside liver cells, it's not going to go there directly. It has to first start from somewhere in the extracellular space, like hepatitis A, for example. It's a fecal oral infection. So you get it by drinking contaminated water. So in the beginning, it's at the surface of epithelium, inside the gut. Then it penetrates epithelium and goes to the blood, comes here. Then it finds the target cell, which is a hepatocyte, and becomes intracellular. So it's not going to be intracellular from the beginning. It cannot be intracellular from the beginning. It has to go through some of the spaces in here, and then here antibodies are present and can target that. Once it's inside cells, then B cells cannot do anything. And then we have to use help from other cells like T lymphocytes, as we will see. So here I have a summary of different paths that pathogens can take. You know, when you travel, you have a starting point, you have a destination, and then you have a path. P-A-T-H, path. So if you want to go to London, for example, England, your origin is Karbala or maybe airport in Baghdad.
Airport.
Your final destination is London. So Baghdad is safe for you. London is safe. But if you're flying over the Russian-Ukrainian territory, where those guys are fighting, your airplane might be shut down. So the beginning is okay. The end is okay. The path in which case. So is the case for pathogens. So some pathogens have to go a long way from their origin to their destination. And all of them have to go through the realm of the antibodies. That is the area of antibodies. So even though antibodies don't have access to this region, before you get there, they can target you, or they can target the infection stage. All right? So this is important to know. So now we are going to look at immune response. I'm going to start from intravesicular space, and then we will move on to intracytoplasmic space. So let's start from things which are inside vesicles. What do we have in here? Well, the first step of defense is very easy. Phagocytosis. They go inside phagosomes, and then there are things named lysosomes that merge with the phagosomes. They become one. And inside lysosomes, there are a lot of toxic stuff. They have acidic pH that can kill microbes. They have something named lysozyme. Lysosome is S-O-M-E at the end. This is lysozyme. It's an enzyme, Z-Y-M-E, that can cut the cell wall of the bacteria. They have proteases that cut microbial proteins, and they have other stuff like lactoferrin that has antimicrobial activity. This is the first step of defense, and it kills a lot of microbes. Is this an innate defense or an adaptive defense? You know, innate immune response, adaptive immune response. Is this innate or adaptive?
Innate.
This is innate. There are no lymphocytes in here. It's innate. These mechanisms have another name as well. Killing mechanisms that don't require oxygen. It means that we have some killing mechanisms later that require oxygen to be used. So this is the first step of defense. What is the next step of defense? Some pathogens like mycobacteria have learned to resist this harsh environment inside the phagolysosome. They live there. They like it there. They're not going to die. They are hard to kill microbes. What happens now? Well, one thing that happens is that their antigens get loaded at the surface of MHC class II. MHC class II is in here. It merges with the phagolysosome. Antigens are loaded at its surface, and it's brought to the surface of the antigen presenting cell for the CD4 T cells to see that. CD4 T cells see it. What happens next? Well, from now on, this cell is named antigen presenting cell. It's showing the antigen to T cells. What happens next? You know, T cells are like sleepy people. Some sleepy people, if you call their name, they wake up. Some sleepy people don't wake up that easily. You call their name, but then you need some stronger stimulus to wake them up. You need to give them a punch. Well, don't do that with your friends, but sometimes people might need some stronger stimulus. So these are like sleepy people. You call their name, they say, yeah, okay, let me sleep for a few more minutes. So you need something stronger. You need to give them a punch. It's called co-stimulation, extra stimulation. And they wake up. What do they do when they wake up? They start producing the cytokine for themselves that makes them proliferate, mitosis. They make copies of themselves. They increase in number. The next step is that another cytokine is produced that tells them what to do. CD4 T cells are also named helper because they help other cells. What kind of help? The help that they provide in here is by production of a molecule named interferon gamma. And T cells that produce that are named T helper 1 cells. We have T helper 1, T helper 2, T helper 1 produce interferon gamma. And they also give a punch back. The friend that you woke up with a punch might give you a punch back. So they give a punch back. What is the punch back? The name of the punch is CD40L, as well as interferon gamma. What happens now is that this cell becomes much stronger. It wasn't able to kill the mycobacterium. Now it can kill the mycobacterium. How is it possible? It generates what is called superoxide. And what is hiding down there, you need to get rid of the passport. What is down there is increased microbial, increased, well, okay, so it doesn't go away. Increased microbicidal activity. Microbicidal. Microbicidal means microbe killing. How does it do that? It produces what is called ROS. Could you write down ROS on your papers? ROS. What is ROS? Reactive oxygen species. ROS. Reactive oxygen species. Oxygen species that can attack other molecules. And they attack the mycobacterium and they kill it. Reactive oxygen species. These are called oxygen-dependent killing because they need to consume oxygen. So this is the process of defense. We have another cell in here which can also help in the process. It's named natural killer cell. Natural killer cells, when they see interleukin 12, they produce interferon gamma to help the macrophage kill the pathogen. The interesting thing is that during immune response, help, if you can call it help from NK cells arrive fast, but it's not very strong. Help from T cells come later, but it's very strong. Why is this fast in a matter of a few hours? This one takes a few days. Why? Because this cell needs to proliferate. Mitosis takes time. And it also needs to change its structure so that it can become T helper 1 and produce interferon gamma. NK cells don't need to proliferate, and they are fast in producing interferon gamma. So that is why these guys are fast, these guys are late. But these guys are stronger because they proliferate. You have many of them. But this one doesn't proliferate. And as well, they have these punches going on between them. They have these connections, the cell surface connections between them. So what we have in here are three mechanisms of defense. 1, 2, 3. One is this innate or adaptive. Phagosome with lysosome. This is innate. NK cells helping macrophages. Innate or adaptive, again innate. T helper 1 cells helping macrophages. This is adaptive, or innate and adaptive together. Innate and adaptive together. Alright, so these are three defense mechanisms. So let's now go to viruses.
I'm sorry to interrupt.
Sure.
But what is the role of the oxygen? What is the role of the oxygen?
What is the role of the oxygen?
Yeah.
The role of the oxygen is that this molecule in here is a very toxic molecule. It's an oxygen with an extra electron in it.
Okay.
Oxygen is very good, but if it has an extra electron, it becomes very dangerous. So these cells need to use oxygen to make this molecule. And the disease that we talked about yesterday, there was a disease named CGD, chronic granulomatous disease. It is a disease that the cells cannot produce superoxide, so they cannot do the killing. It's a type of immunodeficiency.
So we can say it, the cell or macrophage use the oxygen to kill the bacteria.
Yeah, absolutely. Yeah. They need to use the oxygen to produce this toxic stuff.
Only the oxygen, or we also have another molecule?
It's oxygen, and from that you have other molecules too, like hydrogen peroxide is also produced, hypochlorite is also produced, but this is the main factor. This is the main factor. And if you can't produce it, you will have killing problem. You perform phagocytosis, but you can't kill it. And then CGD happens. The chronic granulomatous disease happens if you cannot produce superoxide. So let's go to viruses now. Here I have a virus inside the cell. Imagine that these are 3 cells. The one in the middle is infected. The first step of defense is production of interferons. The previous interferon was interferon gamma, which is produced by leukocytes. Here we have interferons alpha and beta, which can be produced by many cells. The virus infected cell produces interferon, and interferon influences nearby cells and makes them resistant to viruses. How does it do that? And it tries to do the same thing in itself. It's called an antiviral state. How does it do that? It tells them that there is a virus in here, be careful. They say, okay, how can we be careful? It tells them that this virus is coming inside you to make its own proteins. This virus has a lot of proteins. It goes inside the cells to make its own proteins using tRNAs and ribosomes and stuff. It tells them to turn off protein synthesis for a while. Don't make any more proteins. That's the effect of interference. Don't make proteins. If I stop making proteins, it would be bad for me, but it would also protect me from the virus, because virus can't make its own proteins. Interferons are called interferons because they are interfering with the life of the pathogens. Like if you're doing something and I'm interfering with your work, it means that I'm not letting you do whatever you want to do. Interferons block the proliferation of viruses by decreasing protein synthesis. Is this an innate defense or an adaptive defense?
Innate.
This is an innate defense. There are no lymphocytes here. Okay? So what is the second step? The second step is that interferon response is very good, but a lot of times it's not sufficient. So cells put the antigens of the viruses at the surface of MHC class 1. If the antigen is inside a vesicle, like in phagocytes, it will be loaded at the surface of MHC class 2. And CD4 T cells would come to help. If it's inside cytoplasm, it will be loaded at the surface of MHC class 1. And CD8 T cells would come. These guys are killer cells. If they see an antigen at the surface of MHC class 1, they will release their toxic granules to kill. And they also have some molecules that tell the target cell to die. When it dies, all of the viruses inside would die as well. This is like if my hard drive in this laptop gets a virus, well, not a regular virus, software virus. If I cannot get rid of it, what should I do? I would format the hard. So everything is cleared, including the virus. Okay? So what is being done in here, is that it kills the entire cell to get rid of the virus as well. So this is killing by CD8 T cells. So here we have an interesting phenomenon. Where is the whiteboard? Here we have an interesting phenomenon. I'm going to write the name so that you can tell me what it means. So, what is evolution? Evolution. What's the Arabic word for evolution? Takamul. It's the process of gradually making more complex organisms from simpler ones. So what is an arms race? What is an arms race? Or shall I use this? What is an arms race? Oh, this is good. R-A-C-E. What's an arms race? It's related to weapons. Weapons. What is an arms race? Two countries that are fighting with each other, or like India and Pakistan, for example. Americans and Soviets during Cold War. سباق التسلح. So one country develops some weapons, the other one develops better weapons, or ways to defend. Then the next country develops weapons, then the other one develops weapons, and they are continuing this race. It's called arms race. So the process is named host. Oh, that went too big. Sorry. This one. Host microbe evolutionary arms race. What does it mean? Arms race. This one. Host microbe evolutionary arms race. What is the meaning, you think? It means during evolution, we have hosts, it means us, and we have microbes, and we are always challenging each other. Microbes develop ways to infect us. We develop ways to get rid of them. Again, they develop ways to infect us. We develop ways to get rid of them. It's called host microbe evolutionary arms race. So in this example in here, our weapon against the virus is CD8 T cell. What could viruses do to circumvent the defense? Well, some viruses have learned to decrease the level of MHC class 1. If this molecule goes down or disappears, then CD8 T cells will not be able to find the contaminated cell, and they don't kill it. So what is our measure or countermeasure against that? Well, again, NK cells disappear. Host microbe evolutionary arms race. Again, NK cells appear. NK cells don't have antigen receptor, but they have a receptor that detects the presence of MHC molecules, and it's an inhibitory receptor. Inhibitory receptor, most receptors, when they see their targets, they wake up the cell. The cell is woken up, stimulated. This one, when it sees the target, it tells the cell to be calm, go to sleep, don't do anything. So the target is MHC, the ligand is MHC class 1. It checks the cells to see if they have sufficient MHC class 1 or not. If they have it, it goes, yeah, you're a good cell, I'm not going to do anything to you. But if MHC class 1 is down, then it wakes up. Okay, yeah, there is something wrong. Why you don't have MHC class 1? Comes and attaches the cell. NK cells kill the cells that don't have MHC class 1. But before killing, they check for a second signal, just to make sure that they are not killing an innocent cell. You know, inside our cells, we have some antigens, some proteins that are inside the cell. If the cell is under stress, they move to the surface of the cell. These are called stress antigens. Like in here, if I'm stressed, I start sweating and I change, my face changes, and I might turn red. Some symptoms appear in me. So if the cell is under stress, the stress antigens appear, and NK cells check the presence of stress antigens by an activation receptor. So activation receptor tells it to wake up. If you don't have MHC, and instead you have stress antigens, then the NK cell is sure that there is something wrong with you. It releases its toxic granules and kills the cell. So see what happened in here? See what happened in here? First, CD8 cells come to kill. If MHC is down, then NK cells come to kill. By the way, they also produce interferon gamma in the process, just to make sure interferon gamma is to increase the number of MHC molecules. And by the way, this is a big cell, this is a tiny molecule, so you need to have many of them for the CD8s to kill. So if MHC goes up, CD8s kill. If it doesn't go up, NK cells kill. And these are the ways that protects our cells against these things. 3 responses: interferon response, innate or adaptive. Innate or adaptive? It's innate. CD8, killing by CD8, innate or adaptive?
It's adaptive.
Adaptive. Killing by NK cells, innate or adaptive? Innate. These are 3 mechanisms. So now I need you to turn your heads 90 degrees to the right. Can you all do that? More than that, more than that. Okay, so that you can read these things. Alright? So the first one, killing by fungal lysosome formation. The second one,
NK.
NKs help macrophages. The third one, CD4 cells help macrophages. Here is the interferon response. Interferons helping cells protect themselves against viruses. Then CD8s help, and then NK cells help. So these are 6 different mechanisms. So now I want you guys to take a piece of paper or maybe laptops and write down these expressions so that I can make sure that you know it well. 3 mechanisms here. First is this one. Just write it down so that I can make sure that you know it well. The first mechanism against things which are inside vesicles. Formation of phagolysosome. Phagolysosome. And then you will have a break. Phagolysosome. The second one is NKs helping macrophages. This means macrophage. You can say NK help macrophage. NK cells help macrophage. How do they help? By producing interferon gamma. This is the second mechanism. NK cells help macrophages by interferon gamma. The last one, CD4 T cells help macrophages by producing interferon gamma. How about inside cytoplasm? Production of interferons, but these are interferon alpha and beta, 2 other interferons. The next one, CD8 cells killing target cells. And the last one, NK cells killing target cells. Done?
The first mechanism is inside the vesicle.
This one, yeah. This is for this area. This is for that area. Okay, break for 10 minutes. 10 minutes is good?
Yes.
Maybe 5 minutes?
Five.
5? Oh, this is the first time you want to get rid of the class faster, so 5 minutes is better. Okay. Yeah, let's 9 minutes. Okay, so it's 3. We talked about pathogens which are inside cells. Now we need to talk about pathogens when they are in the extracellular space, in here. And here is the realm of antibodies. The antibodies protect this region. Either the pathogens that live in here or the intracellular ones before they enter the cells. That is where antibodies perform their function. So we have seen that antibodies, we have different classes of antibodies, like IgA is secreted into mucosal secretions, IgE kills the worms, and then we have IgG, which is present inside serum. It facilitates phagocytosis and it also activates complement. When a B cell sees an antigen, the first antibody that it produces is IgM. IgM performs some functions, but not all of the functions that we need from an antibody. So what happens now is that if you have a microbe outside cells, some of those microbes will be taken up by phagocytes, and their antigens will be shown to T cells, and some of those T cells become T helper 1. T helper 1s produce interferon gamma. So we had production of interferon gamma when we had something like a mycobacterium that was hard to kill. And then interferon gamma could help this antigen presenting cell produce reactive oxygen species to kill the pathogen. But now the help is needed by B lymphocytes. So interferon gamma tells the B lymphocytes to produce IgG. Because IgG is very important in defense if there is a virus outside of the cells. So that's one way. The other possibility is that sometimes another group of T cells develop which are named T helper 2. How this happens? The antigen presenting cell shows them the antigen, but doesn't give them interleukin 12. If you don't have interleukin 12, you become a T helper 2 cell. And T helper 2 cells produce interleukin 4. And interleukin 4 tells B cells to produce IgE. Because now IgE is needed to defend against worms. Sometimes a third scenario is activated. Let me bring this up a little bit. Sometimes a third scenario happens. If you need IgA, another path is activated. A group of T cells will be generated that some people have called them T helper 3. T helper 3 tells B cells to produce IgA. This happens when the antigen presenting cell is below mucosa, like below gastrointestinal mucosa. So it takes up something, but then it tells B cells to produce IgA, because IgA can be secreted into the mucosal secretions. So this is good when you have something in serum and you want to make it easier for phagocytes to eat it, facilitate phagocytosis, or neutralize it or activate complement. This is good when you want to get rid of something in the mucosal secretions. And IgE is good when you have a worm. We talked about this yesterday. You want to cover the worm with IgE so that eosinophils and mast cells can detect it. And once they detect the worm, they would release their toxic granules, killing the worm. This is the mechanism that it is, if it's activated mistakenly against allergens like pollens or food allergens, then you will have allergy. If IgE is produced against the worm, it's okay. That's where IgE is needed, to kill the worm, to kill the helminths, But if IgE is produced against something which is innocuous, something which is harmless, like pollen that enters my lungs, or against food allergens, food antigens which we eat, and then eosinophils and mast cells come and release their granules, then we have allergy. Food allergy, lung allergy, skin allergy. All right. So these are mechanisms that we know of, T helper 1, 2, and 3. Almost 15 years ago, a new group of CD4 T cells were discovered, and they were named T helper 17. Why 17? All the way 1, 2, 3, all the way jumping to 17? Well, the reason was that there was a cytokine that people had discovered before the cell was discovered, and the cytokine was named IL-17. IL-17 activates neutrophils. But IL-17 was an orphan cytokine. What does it mean to be an orphan? O-R-P-H-A-N? Yatim. Someone whose father or mother has passed away is orphan. How about an orphan cytokine? An orphan cytokine is a cytokine that you don't know who is producing it. The father or the producer is not known. When they discovered this cell, they found that this is the cell that is producing that orphan cytokine. So it wasn't orphan anymore. And they named it T helper 17. You know, in some countries, like in Arabic, it's relatively common. Sometimes the father is named after the child. Like the child's name is Hassan, the father will be named
Ali.
No, not Ali and Hassan. The father will be named father of Hassan, Abu Hassan, for instance. Okay, not Ali in this case. All right. So this is the case in here. This is Abu IL-17. Okay, this is the father of IL, or Ummi IL-17, depending on whether you want to consider it male or female. So this is Abu IL-17, produces IL-17, and it activates neutrophils. So the Abu IL-17 has another child as well. It's named IL-22. But because this is the bigger brother, they name the other ones, it would be about IL-22. So IL-22 is also produced by T helper 17, and it has an interesting effect on epithelial cells. You know, epithelial cells are on the surface of the body. So it tells them that, you know, you are not leukocytes, but you can fight. They say, how can we fight? It says by production of the so-called antimicrobial peptides. They have the ability to produce some antimicrobial molecules. But for that to happen, they need help from T helper 17. All right. So this shows us the structure of the immune system. And you know, this is like an army. Classical armies have a hierarchical structure. What is a hierarchy? What is the name for hierarchy in Arabic? H-I-E-R. H-I-E-R. Where's the white? Hierarchy. Hierarchy or hierarchical structure. What is the word for it?
Tasalsul.
Tasalsul could be hierarchy. You mean that you have different levels.
Yes.
All right. So in classical armies, you have different levels. You have like a central commander. You have the central commander in here, who is a very good soldier. Sorry, it doesn't look very nice. Okay, fights very well, but is also very experienced. Can tell other people what to do. Then you have top officers. Then you have top level high-ranking officers. Like these are the top high-ranking officers. Okay, that are in touch. Sorry, that doesn't look very. Okay. Then you have lower ranking officers. Lower officers. So these are the lower officers. And then you have soldiers. Regular soldiers. And you have many of them. You have so many of them. Could be thousands, could be hundreds of thousands. And then you have ordinary people. You know, ordinary people. Regular people. Ordinary people. So this is the chain of command. Up, down. Central commander, top officers, lower ranking officers, soldiers, and then the rest of people. So, who do you think is the central commander in here? It's the antigen presenting cell. He's a very experienced soldier, fights well, but now he's guiding the rest of people what to do. T cells, CD4 T cells, are the high-ranking officers that are in touch with the antigen presenting cell. And this guy tells them what to do. Become T helper 1, or 2, or 3, or 17. Who are the lower ranking officers? B lymphocytes and CD8 T cells follow the orders from CD4 T cells. We will get there. Not yet. Not yet. Who are the ordinary soldiers? Neutrophils are the ordinary soldiers. You have a lot of them. They fight well. But if you have a guiding officer guiding them, they fight much better. TH17 is the guiding officer for neutrophils. And finally, we have ordinary people, especially people who live at the borders. You know, the borders of a country is the first part that gets attacked. So this officer tells neutrophils to fight well. It also tells people at the borders that you can take up arms, whatever you have, to block the entrance of the enemy. For as long as it's possible, so that neutrophils and B lymphocytes and the rest of the cells can come to your help. That is the hierarchical structure of the immune system. And it's a very efficient hierarchy. So now I need you to turn your heads again, 90 degrees. You see this. If it's in blood or interstitial space, T helper 1, interferon gamma, B cell, and IgG. If it's in here, parasite, T helper 2, IL-4, B cell, and IgE, especially for worms. If it's at the surface of epithelium, Another T cell, some people have called it T helper 3, TGF beta B cell, and IgA, because IgA can be secreted to come to the surface of the epithelium. And here we have two places for IL-17. So if it's in here, T helper 17 with IL-17, and then neutrophils will get activated to perform phagocytosis. What if it's at the surface of epithelium against T helper 17 with another cytokine that it produces it, IL-22, telling epithelial cells to produce antimicrobial entities. So these are different layers of defense. So it's a very complicated structure, but we know that, okay, so the rest of the slides from here, I'm not going to include them in the exam, but it's good if you look at them. The rest of the slides are about evasion of the microbes. Evasion is how to escape or how to suppress. It's part of the arms race. And you can look at them if you're interested. So I have a question here. Exam questions will look like this. Can you answer this question? This is the question. It's asking about viruses in the intracytoplasmic space. So intracytoplasmic, intracytosolic, which one was it? If I come back here, here. Which space was that? It's this space, where viruses live. So the question asks, which of these mechanisms is not a part of defense against intracytosolic viruses? Coming here.
The A?
So which one is not a mechanism of defense? Is production of interferons a mechanism to protect against viruses? Yes, interferon alpha and beta. Is killing by NK cells, lysis means killing, a mechanism to get rid of viruses? Yes. Is killing by CD8 T cells a mechanism to get rid of viruses? Yes. Is activation of macrophages by NK cells a mechanism to get rid of viruses? No. This is a mechanism for things which live inside phagosomes, like mycobacteria. So this is not an example of defense against viruses. All right, so for the exam, your colleagues, your classmates asked, we have the Abu Abbas book. Dr. Abu Abbas is an Egyptian immunologist who's professor at Harvard University, but this book might be too much for you. So the other book is Immunology for Medical Students. Immunology, you can look it up, Immunology for Medical Students. That's a good book. It doesn't have, it's not as complicated as Abu Abbas, and you can read it. For the exam, it would be sufficient to look at the slides and maybe your lecture notes. I don't have anything written to give you except for the PowerPoint file. But doing exam is one thing, learning is another thing. It's always to look at the book, even briefly, to have a better idea of what's going on. But for the exam, questions will be exactly like this. Questions will look like this.
Can you explain more why the correct answer is become D?
Okay, why the correct answer is D. So if I come back here, let me copy this slide, come back here. Okay, so the question is asking about intracytosolic pathogens. Intracytosolic means here. It's asking which mechanism is not effective here. Is interferon effective here? Yes, interferon is effective here. Is cell lysis means cell killing? Cell killing by NK cells effective in here? Yes, cell killing by NK cells is effective here. Is cell killing by CD8 cells effective here? Yes, it's effective here. But whether NK cells activating macrophages is a mechanism, NK cells activating macrophages is not for intracytoplasmic space. It's for intravesicular space. For that reason, that is not the correct answer. All right, so it was an honor to have these classes with you. I hope you do well in your exam and the rest of your medical degree. So we'll be happy to see you for your residency programs at Tehran University. So good luck, and maybe see you later sometime back in Iran.